Good morning, everyone. We're just going to give everyone a few more minutes to get seated, so bear with us. Thank you. All right, is there anything else that you need from me? You're going to be great, don't worry.
Why does my heart feel so bad? Why does my soul feel so bad? Why does my heart feel so bad? Why does my soul feel so bad? Why does my heart feel so bad? Why does my soul?
That's the quick way to jump.
Do you have it added to your schedule? No. Sorry.
Ah, that's the trick. Okay. You guys can talk while we're waiting, so feel free. Well, we'll start up in like 1 or 2 minutes, so. It's gonna be hard to keep track of time since we're off schedule.
Yes,
12 and a half minutes each.
approximately. Yeah. For some of them, if the talks go super long, we've been skipping questions. Why do I feel like everyone can
I
hear
think the mic is
me? Yeah. I was like, I can hear the echo.
Let's talk about the you think she gets started?
Yeah, I think that's a good time.
Okay, well, why don't we go ahead and get started. So welcome to the session on multivariate approaches from methods to applications. I'm Vince Calhoun, and this is Elvisha Damala, and we'll be your moderators today. So it's my pleasure to introduce the first speaker, who's Bo Lee, talking about a high-resolution autoencoder for construction of interpretable brain MRI endophenotypes. So we'll let you take it away.
Thank you. Good morning. My name is Bo Lee. I am a researcher of artificial intelligence and neuroimaging. During the last few days, I have heard that you have problems with your imaging biomarkers. You have biased biomarkers, you have limited size of label data. Well, then it's a time to try some endophenotypes. So here today, I will introduce one of our recent work on the construction of interpretable brain MRI endophenotypes. Specifically, I will introduce why do we need the dimensionality reduction, How can we encode the high-dimensional data into low-dimensional endophenotype? And finally, what can we do with the endophenotypes? What can be decoded there? In population neuroimaging, it is commonly a phenotype of interest that is selected for the analysis rather than using the raw image voxels because of the high-dimensional nature of imaging data versus a relatively small number of observations. However, selecting a suboptimal phenotype, such as specific brain regions, might lead to misleading results or the loss of statistical power, because the underlying etiologies can have far more complex patterns beyond our prior knowledge. So to eliminate the need and the bias for phenotype selection, unsupervised method can be applied. Here we propose an efficient and interpretable method, deep learning algorithm, for dimensionality reduction. Within the category of deep learning, what makes one method differ from another is generally the neural network architecture and the objective function. So autoencoder is a type of unsupervised method, which takes one image as input, encodes it into a compressed feature, and tries to reconstruct the same image from this feature. By maximizing the similarities between the input and the reconstructed image, we assume that this compressed feature can sufficiently represent the input image. Or in other words, the essential information in the original image can be well preserved in this compressed feature. So despite this great success of autoencoder in computer vision field, it is non-trivial to apply such a method in neuroimaging data, because our brain is not as simple as a 2D image of a digit, right? So currently, state-of-the-art autoencoders for neuroimaging are limited to 3D regions of interest or 2D slices only. To solve this problem, a new technique has been developed to train on 2D slices and try to reconstruct the 3D image. Unfortunately, this method did not converge at a high resolution. But their results, however, is very valuable because they show that the bottleneck in this task is not GPU memory, right? Because we are able to fit such a large image into the GPU. And as many of you might know, GPU memory is a bottleneck for deep learning algorithm. So if memory is not a problem, then what is the source problem here? We expect that the challenges underlying this iceberg is the complex 3D pattern of human brain and also the nonlinear spatial relationship across the dataset. Knowing this, we are able to propose corresponding solutions to solve the problem. Specifically, there are 3 key components in the proposed method. The first one is a powerful but also economic neural network architecture. So we empirically optimize this architecture for neuroimaging data, but I will not dive into the details about the optimization. But you will know that this script and the well-trained model will be publicly available. Second is a loss function, or objective function, also known as the loss function. It is the driving force of a deep learning car because the training procedure is basically to estimate a set of parameters that can optimally minimize the value of this loss function. So to encourage the model to learn both intensity and the structural features, we propose this joint loss function that is able to measure the correspondence between the input and the reconstructed image, the similarities between them using a smooth mean absolute error and a normalized cross-correlation. So this formulation of the loss function is new because currently most of the autoencoders are intensity similarity based. So therefore, this addition of structural similarity is very valuable. Last but not least is the pre-processing strategy. What we care about is anatomical variation within the data. But across this neuroimaging data, as you can see from the figure, there is not only anatomical variation, but also a large spatial variation, which itself is a challenging enough task named image registration. We don't want to solve it here, right? Therefore, to encourage the model to focus more on anatomical variations, we apply a spatial normalization as a pre-processing step. We compulated the modulated gray matter maps using voxel-based morphometry, which describe local gray matter density. From population-based Rotterdam study dataset, we included 11,800 3D T1-weighted brain MRI images into this study. After pre-processing, the images are in the MNI standard space, 1 millimeter isotropic, and with background cropped. Okay, finally, we can move on to the results section. First, we evaluate the reconstruction quality of a pre-trained model by applying the model on a separate test set of 1,000 test images. The accuracy of reconstruction is measured by the Pearson's correlation coefficient between intensity of the input and the reconstructed images over 78 brain regions defined on the Hammers Atlas. So in this image, we can see an example of the correlation for right hippocampus, for which the coefficient is 0.71, and the average value over all the 78 regions was 0.77. Secondly, we evaluated the applicability of these endophenotypes to downstream tasks by predicting age using linear regression and predict sex using logistic regression. The accuracy for age prediction was measured by root mean squared error, for which the test performance was 5.14 years, lower than that reported in the relevant work of 6.9 years. In the bottom figure, we reconstruct age-specific modulated gray matter maps, which can be obtained through a backpropagation where the linear relationship between the endophenotype and age. In the colored image, we can visualize the change in brain during aging. Therefore, this region in red can be considered as brain atrophy. Similarly, the prediction accuracy for sex was measured by error under curve, for which we achieved a test value of 0.99, which is high performance, showing the discriminative power of these endophenotypes. And in the bottom figure, we are able to reconstruct sex-specific gray matter images and the changes in brain across sex. So it's also, here we observed asymmetric changes, and also it's worth to note that we don't expect bias of different brain volumes across sex, because these images are linearly registered to a template, therefore their intracranial volume are expected to be the same. And this difference are mainly driven by the local structural changes. With this, I'd like to conclude that to the best of our knowledge, we advanced the state-of-the-art by proposing a first high-resolution 3D autoencoder for brain structural MRI analysis, which is able to encode the essential information in image into an interpretable low-dimensional endophenotype. We have associated this endophenotype with age and sex and achieved high performance at high resolution. We are currently extending this work by involving more target variables, such as dementia, cognition, and genes. We expect that this method and the proposed concept of endophenotypes can be widely applicable in population neuroimaging. With my last slide, I would like to thank all my co-authors and invite you to the poster session. Any suggestions and collaborations from you are very welcome. Thank you.
Thank you very much. We have time for a few questions. So feel free to add questions through the OHBM app. So let me just ask the first one here, which is, how did you choose the architecture for your autoencoder? Like, what was your criteria for selecting that?
The key criteria is to optimize the capacity with a foldable number of parameters. So we did not directly adopt the architecture widely used in computer vision field, but based on my 5 years experience of working on segmentation and registration of brain MRI images. So with less parameter, but higher capacity.
Great, thank you. Have you looked also at variational autoencoder structure?
That's a good point. So as the name says, the difference between autoencoder and variational autoencoder is the variation part. So what we see in the variational part is that they enforce a prior distribution of the learned data, the training distribution. With variable autoencoder, they enforce the distribution of the feature, the endophenotypes, to follow a Gaussian, normal Gaussian distribution, which is not what we want. What we want is to encode the information of each brain image, and therefore to use it for the following prediction, diagnosis. So with variational autoencoder, what they want more is to model normal Gaussian distribution of the training data set. It just reflects the information in the training data set. And as we already heard a lot in the last few days, it is a hypothesis or model-based method. By introducing this prior knowledge, we also limit the ability for discover, exploring power of this model.
Okay, thank you. We'll probably have to move on at this point, but there are a number of questions that maybe you could take a look at later.
Yeah.
People are very interested in this work, and thanks for the very excellent talk.
Thank you.
Okay, so the next speaker is Shami Moore, and she'll be talking about brain age prediction, a systematic comparison of machine learning workflows.
So another brainish talk, but I won't bore you too much. I'm going to talk about a systematic comparison of machine learning workflows in brain age prediction. And I'm a PhD student at Ulish Research Center in third year of my PhD. Yes, so going ahead. Brain-based age prediction aims to estimate person's age based upon data acquired from magnetic resonance imaging. Since brain structure is well known to change throughout age, and there's substantial heterogeneity in how aging affects different individuals, thus, chronological age is not an accurate measure of individuals' rate of biological aging. Instead, machine learning models can be trained on healthy images, and these trained models can then be used to make predictions on unseen sample. For instance, if the true age of a person is 40 years and the predicted age is 42 years, we can then compute the difference between predicted age and true age, which is termed as brain age delta, and in this case, brain age delta would be plus 2 years. But if the true age is 40 years and predicted age is 50 years, then the delta would be plus 10 years. This high brain age delta indicates older appearing brains, and there are many studies which have shown that this high brain age delta is associated with factors of aging and cognitive impairment. Thus, we want to have accurate brain age prediction models. But in order to do so, we need to first understand the effect of different feature representation and machine learning algorithms on prediction error. There are multitudes of studies on brain age prediction. However, they differ in their methodology, and hence they are not comparable. There are different ways in which data can be represented, and there are several machine learning algorithms available. And which of these combinations give the lowest prediction error is not known. Furthermore, many evaluations are performed in rather homogeneous setting where data comes from a single site. But for real-world application, we want to predict data from novel site with confounding imaging effects. And how this combination affects cross-site prediction is not known. Additionally, we want to establish test-retest reliability for these workflows. Therefore, in the current study, we evaluate 128 workflows, which are a combination of different feature spaces and machine learning algorithms to find one best performing workflow. We evaluate them for their within-site performance. We then evaluate them for their cross-site performance, and then we look at test-retest reliability. We then apply the best-performing workflow on clinical data. So these 128 workflows are combinations of 16 feature spaces derived from voxel-wise gray matter data and 8 machine learning algorithms. Out of 16, 12 feature spaces are derived from voxel-wise data with additional smoothing and resampling to different values without PCA, and then additionally applying PCA for dimensionality reduction. 4 feature spaces are derived from Schaefer's, Fan's, and Buckner's atlas with different granularities. 8 machine learning algorithm includes many linear and nonlinear methods widely used in brain age literature. Again, we first evaluate these 128 workflows for their within-site performance. We then select 16 workflows and then evaluate them for their cross-site performance. We then select 4 workflows and then test them for their test-retest reliability. And then finally, we have one best-performing workflow. Coming to data, we use MRI images from healthy subjects with a wide age range of 18 to 90 years from 4 large data sets. We use 3 independent data sets for testing these selected workflows. For pre-processing, we use voxel-based morphometry from CAT 12.8 toolbox, and we use modulated gray matter images as features. Coming to results, first I will go through within-site evaluation results. Here, 128 workflows were trained on 4 data sets separately using 5-fold cross-validation. And now I show the results for CAMCAN dataset from within-site prediction. We see there's a high correlation of 0.94 between true and predicted age, and the mean absolute error is 4.94 years. To find the best performing workflow, we average out the cross-validation mean absolute error over all the folds and then over all the sites. The error ranges from 4.9 to 8.5 years. And the best performing workflow was combination of voxel-wise data, smooth to 4 mm kernel, resampled to 4 mm resolution with Gaussian process regression model. Next, I show the scatter plot between cross-validation mean absolute error and test mean absolute error for all 128 workflows. Since it was computationally very expensive to run all these 128 workflows for cross-site analysis, we selected a few workflows, keeping the variance in the performance, because we did not know how these workflows are going to perform in cross-site evaluation. So these are the selected workflows we use for the next stage of analysis. Coming to cross-site results, here 16 workflows were trained on 3 data sets combined together as training data, and the left outside as the testing data. I show here the example output on CAMCAN data. And again, we see there's high correlation between true and predicted age, and the mean absolute error is 4.88. We then averaged out the test mean absolute error for all the workflows and averaged them over sites, and we found the error ranges from 5.4 to 8.6 years. Again, the best performing workflow was same as what we found from within-site analysis. And now this is the scatter plot between test mean absolute error and cross-validation mean absolute error. We then selected 4 workflows for the further analysis. And these results indicate that the workflows could generalize well on data from new site. Okay, then coming to test-retest reliability results. Here I'm just showing results with one best performing workflow. So here we trained the models using all the 4 data sets combined together as training data. Since brain morphology is not expected to change within a short span of time, we expected brain age delta to be also stable within a short duration of retest duration time. The test-retest reliability was measured using concordance correlation coefficient. We tested this on 2 independent data sets. First, we looked at core data set, which had 86 subjects scanned twice within a retest duration of 3 months. And we found there's a high correlation of 0.97 between brain age delta from scan 1 and scan 2. Next, we looked at OSS data set, and we found similar results. With this, we can say that brain age estimation is stable within the same subjects across the short delay of 3 months. Next, we apply the best performing workflow on the clinical sample. Since there's higher atrophy associated with neurodegenerative disorders, we hypothesized that there would be higher difference in true and predicted age in clinical population as compared to healthy controls. And we tested this using ADNI data by comparing brain age delta between cognitive normal, early MCI, late MCI, and Alzheimer's patient. We found that there is significant difference between brain age delta between Alzheimer's disease and every other group. We also performed age bias correction in the predictions for all the group using control group following the standard methods. These results indicate that higher brain age delta in MCI and AD, and it indicates that it's associated with accelerated aging. Next, we investigated associations between delta and various clinical test scores commonly used to measure disease severity in dementia. First, we looked at mini mental state examination. Lower value indicates higher impairment. And we found significant negative correlation between MMSE and delta in late MCI group and Alzheimer's group. Next, we looked at functional assessment questionnaire. Higher value indicates higher impairment. And we found significant positive correlation between FAQ and delta, again in LMCI and ED group. We did not find any correlation in cognitive normals and early MCI patients. So these results indicate that high brain age delta is associated with higher disease severity and cognitive impairment. So with these results, we can come to following conclusions. First, there's effect of both feature representation and machine learning algorithm for brain age prediction. Second, we found voxel-wise data was better than parcel-wise data for prediction. Brain age delta is reliable over short duration of scan interval. And we found higher brain age delta in late MCI and Alzheimer's group, which was also associated with higher cognitive impairment. And with this, I would like to thank you for your attention. I look forward to seeing you at my poster. And I would also like you to visit our poster for ULearn, which is the library I have used for my analysis. Thank you so much.
Thank you very much. First question is, are the brain age gaps consistent across the different workflows?
So there is some variation of errors. We see that the errors is between 5 to 8 years, which doesn't mean that there's just one-to-one correlation. But if I correlate these predictions, they are highly correlated to each other, ranging from 0.8 to 0.9.
But that's the MAE, right? Did you also look at brain age gap?
So, yeah. But
Okay. Both of those are similar?
Sorry?
Like the brain age gap versus just the age prediction? The delta?
I didn't get your question.
So the difference between the chronological age and the
True age, yeah.
true age, so that's the area you're talking about, it's the 5 to 8 years.
The delta, yes. It ranges
Yes.
from plus to minus 5 years, yes.
Okay. Another question, did you optimize hyperparameters or?
Yes.
Okay.
So we did everything in the cross-validation consistent manner, and we had a nested cross-validation for hyperparameter tuning for all the models, wherever it was necessary.
Great. Next question, did you check the bias size if you're training with one site and testing with another site, just how large that was?
Yes. So we saw that for within-site analysis, we could easily correct for bias correction. But when we go from cross-site analysis, and if we do the bias correction using the training data, it doesn't work very well. And that's why when we went to the clinical data, we used, I'm talking about the ADNI data set, which I showed here. And there we did the bias correction using the cognitive normal subjects from the same site so that we have a better bias correction.
And I think we probably need to move on at this point. Again, there's a lot of questions, so jump in there, and I'm sure everyone would appreciate your interaction. So thank you very much.
Thank you.
Okay. And our third talk is by Florence Allen, and the title is Convolutional Neural Networks Predict Outcome from Coma Based on Auditory ERPs.
Okay. Sorry.
Okay, you can take it away.
Yes, so thank you for the introduction and also for the opportunity to present my work here. Yes, okay. So cardiac arrest is one of the leading causes of coma and admissions of patients to the intensive care units of hospitals. It is also the cause of around 17 million deaths every year worldwide. Early medical intervention has a strong impact on patients' chance of survival, and for clinicians, it is very important to make an early prediction of patients' chance of patients' outcome for informing families and also making a precise decision about when to stop life support. Once patients are admitted to the ICU, they immediately undergo a controlled temperature management at either 33 or 36 degrees Celsius. In the following, I will call a temperature management at 33 degrees hypothermia and at 36 degrees normothermia. During their stay in hospitals, clinicians perform a multitude of evaluations on the patients, one of them being EEG recordings. Yes, until either patients they die or they recover or the life support is stopped. The later is decided by the clinicians based on a multitude of current markers, most of which are related to or are based on visual evaluations, such as the example here of an EE recording and a clap performed at the bedside of the patients. We can see a decrease in the amplitude of the EEG after the clap, indicating a reactive EEG background and therefore being a marker for good outcome. We selected 4 currently used markers, and if those markers did not agree on the patient's outcome, we would call the patients to be in an uncertain state because the clinicians would not be able to make a precise prediction. We call these patients to be in a gray zone. Currently, clinicians have a lot of markers for bad outcome and only a handful for good outcome. Most markers related to good outcome are related to auditory stimulations. So for example, here on the right side, we have a patient with an N100 reaction to an auditory stimulation, whereas the patient on the left side does not have any such reaction. The presence of an N100 can be an indication of a good outcome. However, these studies were performed at later stages of coma and only on single electrodes. More recent attempts looked at multivariate decoding algorithms, where they trained a machine learning algorithm per patient on the EEG data and also found a marker for a good outcome. And then in recent years, obviously, computer vision and deep learning have gained quite a lot of attention for classifying images, videos, and a multitude of other things. They have been used for decoding EEG for quite some few studies already, and they have also been used for predicting outcome of coma based on resting state EEG recordings. Here, however, we hypothesized that convolutional neural networks would be able to extract patterns of EEG responses to standardized auditory stimulations, and that the networks would improve outcome prediction for patients in an uncertain state. We are using EEG recordings of patients on the first day of coma after cardiac arrest, being exposed to standardized auditory stimulations. We have 79 survivors and 55 non-survivors. The data was recorded in different centers and under different protocols across Switzerland. We have, for example, 79 patients treated with hypothermia and 55 patients treated with normothermia. Each patient is exposed multiple times to these auditory stimulations. So for the training of the network, we feed each of those trials through the network to get a prediction between 0 and 1, indicating if that trial was more likely to belong to a survivor or non-survivor. Survivor being 1 and non-survivor 0. Then for testing, or at the end of the training, we take all of the trials per patient and run them through the network and take the mean of the predictions of the network to get one value per patient, indicating if that patient was more likely to be a survivor or non-survivor. The network architecture that we're using is called EEGNet and was a network developed specifically for EEG decoding. We run a tenfold cross-validation for the training. For visualization purposes, I will only show results of the best fold here. This graph shows the output prediction, the mean output prediction of the network per patient for survivors on the left and non-survivors on the right side in orange. We also depict separately train and test and validation sets. You can see that we reach an AUC score of 0.83 and a positive predictive value of 0.92. Meaning that if a patient had a value above 0, network output value above 0.5, they were more likely to survive. However, if that value was below 0.5, we cannot make such a strong claim. Next, we look at the network performance on these gray zone patients. So the patients in an uncertain state. We had 48 patients, so roughly a third of our patients. We reach a positive predictive value of 0.86, which is slightly lower than for the full cohort, but still quite high. So we have seen so far that the networks are quite good at predicting outcome from coma based on these auditory stimulations. However, we also wanted to explore what the network would learn. A recent study in our group looked at the phase locking value in hypothermia patients. The phase locking looks at the phase difference between two electrodes and is therefore related to neural synchrony. This study showed a positive predictive value of 0.87 for the phase locking value, and patients with a high phase locking value were more likely to survive. We did reproduce, first off, these results in normothermia patients, where the positive predictive value was slightly lower. And then we looked at the correlation of the network output and the PLV, where we found quite a high correlation, indicating that the network seems to pick up some of the features related to the PLV. So if a patient was, according to the network, more likely to survive, they also had a higher phase locking value. And if they were less likely to survive, they had a lower phase locking value. We also depict some exemplar EG traces of survivors and non-survivors, both correctly and incorrectly predicted ones. The same study I mentioned before also looked at the Lempo-Sif complexity in hypothermia patients and did not find any indication of outcome prediction for this complexity. The Lempo-Sif complexity measures the complexity of a signal and is related to neural complexity. We did reproduce these results in normothermia, also not finding any indication of outcome prediction. However, if we look at the network confidence, again, versus the Lempo-Sif complexity, we also here find quite a high correlation, indicating that the network also seems to pick up some of the features related to the Lempo-Sif complexity. For patients with a high network confidence had a rather low Lempo-Sif complexity, and patients with a high confidence of the network had a higher Lempo-Sif complexity. So I have shown two examples of electrophysiological measures that are related to the network's output or prediction from coma and are therefore features picked up by the network. So in conclusion, I've shown a high predictive power of convolutional neural networks on predicting auditory ERP data, a generalization across cohorts treated with different temperatures, a high predictive power for a subgroup of patients in an uncertain state, and the network confidence correlates with the phase locking value and the Lempo-Sif complexity, giving us some insight into the network, and the presented pipeline could be used in a clinical routine as a positive predictor in the future. And with that, I'm at the end of my presentation. I'd like to thank the members of our group, especially Professor Atina Tsavara and my collaborator Sigurd Alnes, as well as the funding resources and all of you for your attention. Thank you very much.
Thank you very much. One question that came up was you showed results from, I think, the best fold, you said?
Yes.
And so that, of course, is just one snapshot and sort of a best case scenario. So did you look at how the results compared to the other folds or on average, et cetera?
Yes. So because we show like a lot of measures related to exactly one patient, we look at the best fold, but of course we evaluated all folds, and the results seem to be pretty consistent across all folds. Okay. No matter if the patients were in the training or the testing data set. Okay.
Another question was, since this is time series data, yeah. Instead of CNNs, why not try something that looks at sequencing, like RNNs or LSTMs or other types of temporal models?
Yes, so the convolutional neural network that was used here was designed specifically for EG data. So it does convolve the data in a time-wise manner, first across the time dimension and then across the channel dimension, so taking the time into account in some way. But yeah, of course, it would also be a possibility to look at different time-related architectures, yes.
Okay, thank you. And then which electrodes did you use in your analysis? All of them or some subset, or if so, which ones?
So the data was recorded from 2009, I think, to 2017, and early recordings used 19 electrodes, whereas later recordings used 62 electrodes, so we only selected the overlapping set of 19 electrodes here. Yes.
Okay. Thank you very much for the excellent talk.
So our next talk will be about optimizing analysis choices for multivariate decoding by creating pseudo-trials using trial averaging.
Hi, good morning. So this is work that I did while I was a postdoc with Alexandra Walger at the CBU in collaboration with Tal Gruiswagers at the University of Western Sydney. And we collected a lot of MEG and fMRI data in the lab, and we ran a lot of decoding analyses on this data. But as you all know from the talks you've already seen, there are a huge number of choices that you have to make in how you run your decoding analysis. And so we're really going right back to basics now. We really wanted to test a number of these different parameters and see how they would affect our decoding analysis. So, of course, the aim of your classification is you might take a sample of your data, as shown on the left, with, say, 2 conditions, shown in different colors here, and you're training a classifier to separate between these 2 conditions. And then you might apply this classification on some unseen data, and you can calculate your favorite metric of choice to see how accurate it was at classifying between those 2 data sets. Of course, the assumption being that if you can classify between these points, then you have some meaningful information in your brain signal about these 2 conditions. But if you're presented with a large number of trials, so for example, if you have EEG data, you then have the question of whether it's better to provide your classifier with fewer, more stable estimates, so for example, by averaging over your trials, or by giving your classifier as many data points as you have. So in this case, you might reduce your trial numbers by averaging over them, or in this instance, you might keep all the trials as they are and give them to the classifier. And it was kind of unclear to us which of these would be the most optimal in different experimental setups. So for example, depending on your effect size, depending on how many trials you have, depending on how many cross-field validations you want to do, and depending on the type of classifier that you might want to use. So we set out to test a number of these using simulations. For the sake of time, I'm just going to show you one set, but we do have more in the paper that should be out shortly. So what we're doing is we're using a Toolbox Cosmo MVPA to generate some very simple simulations. In this case, we're just simulating 2 different experimental conditions with a medium effect. And what we chose to do was simulate 90 trials that, in this case, we've split up into 3 blocks or chunks. Of course, this is also a decision that will influence your decoding, and we do explore that more. And we tested a couple of different classifiers. Just to orient you to the graphs that I'm going to show, down on the x-axis is showing how many of our trials we've included in an average or a pseudo trial. On the x-axis, we have our % decoding accuracy. We're also showing a t-score against chance decoding, which in this case, of course, is 50% accuracy. So let's take our naive Bayes classifier, and we're going to start with 1 trial per average, which is obviously no averaging. And you can see that at the baseline level, we've got a 55% decoding, which is pretty good, and a t-score of 11. Now, if we fill in the rest of this graph, what you can see is that even by including 2 trials in our average, we've already boosted our decoding accuracy, and sort of more crucially, perhaps, we've increased our t-score in detecting against chance level. However, particularly for the naive Bayes classifier, you can see that you don't want to just keep increasing the number of trials per average. As you can see, when we've included more than 15 trials per average, which is over half the number of available trials per block, we have this dramatic reduction in the classifier accuracy. And so for this classifier, which you might expect based on how it works, it prefers to have less stable estimates of the trial rather than giving it fewer averages. Now, of course, we're not the first people to show that averaging is beneficial, but we just wanted to demonstrate this across a wide number of parameters. Again, just in case it wasn't clear, just to point out the difference between these points on the graph here. Of course, if we do no trial averaging, then at the first point we can give 90 samples to our classifier. But once you start increasing the number of trials per average, you're obviously reducing the number of trials that you're giving to your classifier. So for example, if we're averaging 5, then we do 90 into 5, so we're giving the classifier only 18 samples, right? Now, kind of crucially with the simulations, we just wanted to point out here that we're not artificially creating an effect with our averaging procedure. So here I'm showing you where we've simulated no effect, and you can see that the accuracy is hovering around chance, and our t-score is relatively low here. Another really important point here is that these results are relying on the principle of creating what we're calling pseudo-trial sets, but it's just an iteration procedure. So what we do is we randomly allocate our trials into these averages 100 times, just to make sure that the accuracy is not dependent on some random allocation of some of the trials. We have played around with that parameter as well, and we found that 100 iterations was a good number really to stabilize this effect. And just to show you one example, if you don't do this, if you just do it once, if you allocate your trials once to the pseudo trials rather than the 100 iterations, you obviously have fewer possible outcomes, and there's sort of a similar drop in the t-score, but a maybe more severe drop here. Now you might be thinking, well, I don't use naive Bayes, what about other classifiers? And again, as you might expect, because the maths and the assumptions are different, they have different trends in the effective number of trials per average. So here is a SVM classifier as implemented in Cosmo MVPA. And you can see this initial jump. So, some averaging is beneficial. We're increasing our decoding accuracy and our t-score. But really, it was unnecessary to do more trial averaging in this case. And a similar trend for LDA. You can do more averaging. You have a fairly stable decoding accuracy. However, you didn't really benefit much from the extra computation time. So the other thing that we really wanted to try, which is implemented in Cosmo MVPA, and we weren't sure how it would influence our decoding, was this method called resampling, which, as you would expect, really simply means that you just allow your classifier to use the same trial multiple times. So you can create more pseudo trials, more averages, because you're using the same trial in several different averages. And we thought this might be beneficial because it would help us in this trade-off between being able to provide stable estimates by averaging, but also having a large number of samples to give to our classifier. So we also tested along this dimension. Here, what I'm showing you on the left is the SVM classifier, the graph that you've already seen with no resampling. And on the right is what happens when you allow each trial to be used twice. So essentially, you've doubled the number of trials that you can give to the classifier. And what you can see, there's not a huge amount of difference between these two graphs. So we've increased the standard deviation across our 100 participants or simulations. And there's a slight stabilization in the t-score, but really, we didn't see a huge benefit, at least in our simulated data. And just to show you a bit higher along the graph, now I'm resampling at 5, but still a fairly similar outcome. And then again, just to really point out, if I wasn't clear the difference between these two simulations, of course, on the right, if I'm including 5 trials per average, but I'm resampling by 5, it means I'm back to my 90 trials per decoding simulation. Just to show you the whole space that we tested across, and now I'm showing the number of trials per average on the left, on the x-axis here, and you can think of that very first column is basically another way of visualizing the scatter plots that you already saw. And then down on the x-axis, is the number of trial resamplings, so the number of times we're allowing a trial to be used. And of course, we've got our mean decoding accuracy, our standard deviation, and our t-scores presented here. Now, if you're just looking at the mean decoding accuracy, as we saw, there is a slight improvement as you increase the number of trials. There's not a huge difference shown by the trial resampling. But again, the more you increase the number of trials per average, the more you increase your standard deviation, and so that has a negative effect on your t-statistic. So really, you kind of need to be, or only need to be in the lower left-hand corner of this space to optimize your decoding in our simulations. Again, just to highlight the different classifiers, LDA has a very similar trend. So you're increasing the decoding accuracy with more trials, but you're increasing the standard deviation, and resampling doesn't really help you in terms of your t-statistic. So again, you only really need to be on the lower bottom left side of this curve. Of course, as we already saw, naive Bayes performs slightly differently. It has different assumptions, of course. And so again, you know, minimal averaging and minimal resampling is sufficient really to find the effect that you're looking for. Here is all of the data put together, just so you can see the comparison. And yeah, just to summarize again, that a small amount of averaging was helpful for your classification, but we didn't find a really strong benefit in the resampling here. So of course, there are a huge range of parameters. I've only shown you a few in the sake of time. We do explore a lot more in the paper. And generally, across all of these different simulations, we found the general decoding approach would be to create averages between around 1/4 and 1/2 of your available trials. That seems to work well, at least across all the parameter spaces we've tested. So different effect sizes. We did find if you have a slightly lower effect size, then you might want more averaging, which is why I've said between 1/4 and 1/2 here. And you can use resampling, but it wasn't hugely beneficial above high values. And it obviously takes much longer to run your analysis if you're giving the classifier more and more data points. So of course, as you might expect, there's this clear trade-off between providing the classifier with fewer noisy trials, but more cleaner trials. Sorry, the other way around. But this does seem to vary a lot with the choice of classifier and your underlying effect. And we do explore more of these in the manuscript, which should be out on bioRxiv once I've finished writing it. Thank you for listening. I do have a poster today, so if you have any questions that aren't answered in the session, then do come and chat to me about it. Thanks.
Great, thank you so much for an excellent talk. So there's a few different questions sort of asking, why do these approaches help if it's not necessarily adding new information?
Well, averaging would help for obvious reasons, right? Because your single trials will have more noise in them. So, you know, just by averaging a few of them, you've, in theory, reduced the amount of noise in your signal, right? Assuming that the signal is stable across trials. And resampling, as I said, was really just a way to increase the number of trials that we could give to the classifier, assuming that we were assuming it might help, and it didn't actually help as much as we thought.
Great, thank you. And another question, or another theme that's emerged is, you've shown results from a few different models. How do you think more complex deep learning models might compare?
Yeah, so this was really simplistic starting point work, just to sort of ask some questions about these parameters. Of course, adding more complexity would always help us find more information. Yeah.
Great, thank you so much. And for the next one, unfortunately, the speaker could not be here with us, so we're going to be playing the video. Thanks.
Hi everyone, I'm Dong Meizhi from Beijing Normal University. I'm currently working with Professor Jin Sui. Now I'm going to talk about my project using resting-state functional connectivity to identify depression subtypes with higher ECT response rates. In this talk, I will briefly talk about the background and the problems, then describe the method adopted to solve the problems. Finally, I will demonstrate some key results and conclude my talk. As is well known, depression is characterized by persistent anhedonia and emotion deficits. Depression is a largely heterogeneous mental disorder driven by varying clinical symptoms. And depression is often comorbid with other mental disorders, such as generalized anxiety disorder. Most importantly, depressive patients often respond differently to a given specific treatment or intervention. The large heterogeneity of depression seriously hinders precise diagnosis of depression, which may delay timely and optimal treatment. Moreover, the number of depressive patients is increasing year over year, causing severe social and economic burden. So our challenges are how to objectively pass the heterogeneity of depression and seek the optimal treatment for each subject. Recent studies have tried to find the best therapeutic targets and pass the heterogeneity of depression using brain neuroimaging. For example, functional connectivity between subcallosal cingulate cortex and the above three regions were associated with remission to antidepressant. Specifically, the remission rates of an individualized treatment can achieve at 86.4%, targeting at the regions of left dorsolateral prefrontal cortex, most anti-correlated with subgenual anterior cingulate cortex for each participant. Additionally, 4 subtypes of depression were identified by combining functional connectivity and depressive symptoms, and showed different response to rTMS treatment. And 6 brain circuits were defined for depression and anxiety, and can distinguish response to antidepressant and behavioral intervention treatments. Taken together, the data imply that functional connectivity can partly pass the heterogeneity of depression, showing greater potential application in individualized treatment. In this study, we aim to use functional connectivity to pass the heterogeneity of depression and identify replicable and generalizable depressive subtypes. Further test whether distinct subtypes have different behavioral manifestations. Most importantly, we explored the specificity of each subtype in response to ECT treatment. First, whole brain functional connectivity was estimated for each subject based on AAL atlas. Then, we used non-native matrix factorization to divide all participants into subgroups in the discovery and replication datasets respectively. Non-native matrix factorization is a statistical approach that decomposes data into non-native parts and allows us to simultaneously cluster features and individuals into subgroups. A particular advantage of path-based representation of NMF is that it can mine similar aberrant patterns within only a subset of groups, and such nonlinear relationship cannot be detected based on global similarities. In 2 independent datasets, 3 clusters consistently showed an optimal fit of the data with high stability, which were dominated by frontal-parietal, contralateral, subcortical occipital connectomes, and a high spatial correspondence was observed for the corresponding subtypes, with dice similarity coefficients of 0.81, 0.81, and 0.78 respectively. Similar results were observed at node and network levels. Subtype 1 were represented by connectomes among default mode, lateral attention, frontal-parietal, and sensory motor networks. Subtype 2 were represented by connectomes within contralateral networks, and subtype 3 were represented by connectomes between subcortical and visual networks. We further conducted a neuroscience meta-analysis to seek the representative behavioral topics associated with connectomes of each subtype, suggesting that each subtype was linked to different behaviors, namely listening perception, face recognition, and episodic memory, and reward anticipation respectively. Additionally, a linear support vector massif was used to test the discrimination of the 3 subtypes within and across datasets. Results showed that SVM yield high accuracy of 89% and 80% when classifying 3 subtypes within each dataset, and 86% and 83% for the across dataset prediction, suggesting high replicability of the derived subtypes from adaptive data class 3E. Compared with Hessian controls, subtype 1 showed hyperconnectivity between default mode and ventral attention networks, between default mode and sensory motor networks, and between frontal parietal and sensory motor networks, especially the prefrontal parietal connections. And subtype 2 showed prominently decreased connections between frontal parietal and visual networks or sensory motor networks, and between subcortical and visual networks, especially the striatum occipital prefrontal hyperconnectivity. For subtype 3, increased connections were observed between subcortical and visual networks, while decreased connections between limbic and sensory motor networks, and between visual and sensory motor networks. After obtaining the cluster-defining features for each dataset, the cluster signatures for each subtype were finally denoted as average connections of the overlapped cluster signatures across datasets. Most importantly, to identify the most treatment-responsive depressive subtype, we mapped unseen individuals receive ECT treatment into their corresponding clusters using the cosine distance of cluster signatures to the closest centroid. Results demonstrated that the response rate of the subtype 2 was higher than the other 2 subtypes with ECT treatment, while subtype 1 showed the lowest response rates, implicating that depressive patients classified as subtype 2 could be more likely to recover after ECT treatment. And here is the conclusions. We identified 3 reproducible and generalized subtypes for depression across datasets, which were represented by frontal parietal, contralateral, and subcortical occipital connectomes respectively. Different subtypes were associated with distinct behaviors, namely listening perception, face recognition, and episodic memory, and reward anticipation respectively. Especially, the frontal parietal connectome subtype had the lowest response rate, while the subcortical occipital subtype had the highest response rate to ECT treatment. Finally, for this project, I would like to thank my advisor Jinsui and all collaborators. Thank you for listening. If you have any questions, please feel free to email me.
So we won't be able to take any questions for that one, but please do reach out via email if you have any questions. And last but not least, we have Hamid Karumi Ruzbahani here to talk about spatiotemporal analysis of information exchange in multiple domain network using fMRI MAG fusion.
Hello everyone, thank you for coming to my presentation. Today I'm going to present my work which is on understanding and characterizing the multiple domain network. So the multiple domain network is a set of brain regions which expands across frontal, parietal, as well as occipitotemporal areas, which become active across a wide variety of tasks, especially when the task is difficult. So the level of information processing in the multiple domain regions changes with perceptual difficulty, task relevance, attention, etc. As you can see in this figure, when the perceptual difficulty of the stimulus was higher, the encoding of that perceptual information was higher in the multiple domain network. The most recent hypotheses about how what the multiple domain network does is that it might connect different parts of the brain to one another, different systems such as the sensory areas to motor areas and high order cognitive areas to one another as a kind of platform for information exchange across the brain, connecting different functionalities across the brain. However, this hypothesis has not been tested yet because most of the studies of the multiple demand network have used functional MRI, which does not give you enough temporal resolution to be able to track information flow across the brain at high speed. As well as the other thing which has remained unclear is whether there is any information exchange across the multiple demand network. There have been some functional connectivity analyses across the multiple demand network which have found connectivity, but we are not sure whether that connectivity is only activity or if there is any information exchange across the brain. So in order to see whether there is any connectivity across the multiple demand network and whether that connectivity contains any information, we used a stimulus response mapping paradigm in which we had 4 stimuli. We presented one of 4 stimuli to the participants, as you can see here, and they had 4 buttons to press, one button for each stimulus. So, and the mapping between the stimuli and the buttons were called rules, and they were indicated by the color of the fixation on the center of the screen. As you can see, the structure of a single trial here. On each trial, the participant saw the presentation of one of the 4 stimuli and the fixation which cued the rule or the mapping that they needed to use. And they learned the mapping between the stimuli and the rule in the training session before the actual scanning session. So we decoded 4 aspects of information from the brain activity in both MRI, fMRI, and MEG. The first one was the coarse stimulus information, which we defined as the discrimination between left stimuli, the 2 left stimuli, and the 2 right stimuli. So because they are easier to discriminate from, these pairs are easier to discriminate from one another, easy to discriminate from one another, both perceptually and potentially on the brain activations, we call them relatively easy or easier aspects of stimulus information or coarse stimulus information. There was another aspect of information which we call fine stimulus information, which refers to the discrimination between inner stimuli from outer stimuli. And as you can see that those 2 aspects, inner versus outer stimuli, the discrimination of the 2 would be more difficult because the stimuli have overlap with one another. So in both brain data as well as perceptually, it should be more difficult. And we also decoded rule information, rule 1 versus rule 2, the different mappings between stimuli and the responses. Also, we had response information which we decoded, inner fingers versus outer fingers. So going to the results, in fMRI decoding, we've decoded the 4 types of information, as you can see here. What we saw was that, as expected, stimulus information, both coarse and fine stimulus information were high in visual areas as well as lateral occipital cortex, and it was lower in the multiple demand network. But interestingly, while the coarse stimulus information was higher in the visual areas, as you can see, they were lower in the multiple demand network. And as I mentioned, the fine stimulus information was the harder or more difficult aspect of stimulus information. And the fact that it is more pronounced across the multiple demand network may suggest that the multiple demand network seems to be become active and more involved in the processing of the more difficult aspect of information compared to the coarse stimulus information. We also saw rule information across the multiple demand network except for AIFO here. And also the visual area, and we could also decode response information from IPS, as you can see here. So going to the MEG results, we observed that so here we decoded across regions of interest, but here in MEG data, we decoded the same information, the same 4 aspects of information from MEG across all channels, aligned to the stimulus onset time and as well as response time. So these 2 are the same data, but aligned differently to the stimulus onset as well as response onset. You can see that after stimulus onset, course stimulus information was much higher and above chance, and higher than the final stimulus information after the stimulus onset. But interestingly, they swap right before the response, with final stimulus information being higher than the course stimulus information. And rule and response information were decodable, not mostly aligned to the stimulus onset, but rather aligned to the response of onset, very close to the response. So based on these results, we may infer that the effects that we can see here with the domination of harder aspect of information being dominant in the multiple domain network, and it showing the same effect in the right before the response, we might conclude that the multiple domain network might be the reason for that specific domination of harder aspect of information. But we cannot make that conclusion based on fMRI or MEG alone, because they give us information about where and when the encoding of information happened in the brain, but not the intersection of the two. So we cannot tell where and when things happened in the brain. In order to cover that point, we fuse the two modalities using fusion analyses. For that, we calculated the correlation between fMRI representations and MEG representations, and a model which determined what aspect of information we were looking for. So this has been developed before, and based on so these are basically correlation of the two modalities, fMRI and MEG, across time and across areas, as you can see from the seminal study in 2014, that based on the timing of these patterns of correlation, they infer information about where information appeared first and where it moved to later on. So you can see this study. We did the same approach using representational similarity analysis. We came up with matrices from all sensors in the MEG and across every time point, and every region of interest in MRI across all boxes within that region of interest. So we structured these different RDMs, representational dissimilarity analysis, which allowed us to combine MEG and fMRI. And then we obtained the time courses of information correlation between the two modalities. So for course stimulus information, what we found was that there seems to be some gradual shift of information across the visual area to LOC, IPS, AFO, IFS, and ACC, which are from posterior to anterior parts of the brain, from visual areas to anterior parts of the multiple domain network. Based on the peaks of information, you might conclude that information seems to be going from the back of the head towards the frontal part of the brain. And we saw somehow similar results for rule information, which showed that information appeared first in the multiple domain network, and then you can see the peak of information, of rule information appearing in visual areas. So it might suggest that these time courses may suggest that rule information appeared first across the multiple domain network and then moved back towards the visual areas. So these are what we should conventionally infer from the fusion analyses. Final stimulus information also shows feed-forward information, suggested feed-forward information based on the timing of the timing when they reach the maximum correlation. And response information showed the latest time point of correlation at IPS, which was where we saw the most information about IPS. However, these results do not give you quantitative insights about whether any information is exchanged across the brain or not. To cover that, following on the recent shift from univariate analysis of connectivity to multivariate analysis of connectivity, we made use of the commonality indices, or the correlation between the two modalities, the time traces that I just showed you, to see if there is any statistical relationship between the time courses across all the areas that we had in our analysis. So we tracked the information from the source area to all the destination areas, from all source areas to all the destination areas. And we had 6 regions of interest. We tracked the information across the 2. And what we saw was that these are summarized here. You can see for stimulus-aligned data and response-aligned data. And I'm showing these significant results in red boxes, but more clearly here. So we can see that for coarse stimulus information, we had no above chance connectivity across the brain from visual areas to lateral occipital area or any other parts of the multiple demand system. And however, for the fine stimulus information, which was the harder aspect of visual stimulus, we clearly saw connectivity or information flow from occipital, from different parts of the multiple demand network back to the visual areas aligned to this after the stimulus onset, and from visual areas to the ACC area before the response was made. So we can see clear connections between different parts of the multiple demand network and visual areas, which is not the case for coarse stimulus information, which was an easier aspect of stimulus information to process, and that might be why the multiple demand network did not get involved. And rule information also showed more information transfer aligned to the response, mostly going towards the ACC area, and response information was mostly fed back to the posterior areas from the ACC area. So based on these results, we can use MEG or EEG fusion with fMRI to obtain insights about when and where information is represented and transferred across the brain. We saw that the fusion itself showed some gradual shift of information from the posterior brain areas to anterior brain areas for stimulus information, but through connectivity analysis, we did not see that any statistical relationship between those areas. And rule information also, based on the fusion analysis, we saw that it might develop in the multiple demand network and feed back to the visual areas, but connectivity analysis showed that it is mainly response aligned, and it goes to the ACC area, which was not clearly observable in the fusion analysis alone, but through the connectivity, yes. And also response-aligned information appeared in the IPS area before the response, and it was dominantly received by the ACC area around the stimulus onset and fed back around the response time. So conclusions would be the temporal dynamics of information processing in the multiple demand network showed that different nodes of the multiple demand network become active across different parts of the trial, and not all the nodes contribute to the processing of the same information and with the similar dynamics. And you can use this proposed informational connectivity analysis, which is based on fMRI MEG fusion, to obtain insights about how different parts of the brain interact with one another. And I would like to thank you for your attention and my collaborators for their contribution to this talk. And I'm happy to take any questions you might have. Thank you.
Great, thanks for such an amazing talk. So we'll take 1 or 2 questions. Something that's come up is, how does the multiple demand network differ from other networks that have been implicated in this? So like salience, dorsal, ventral attention.
There are some overlaps between the multiple demand network and the dorsal attention network. And I think the multiple demand network has more nodes, and it is more scattered across the brain, and it has been shown that it becomes active across many different tasks, such as even mathematical calculations, working memory, and different tasks which might not necessarily activate the dorsal attention network, based on my understanding.
Great, thank you so much. We are a few minutes over, so we'll wrap it up. But if you have any questions, please do come up to the authors, check out their posters, and thank you so much for joining us today.